Namaskar. My name is Pranjal Pratim. I am working as a faculty in the Department of Biotechnology in Darrang College, which is under Guwahati University. So today I am going to deliver, discuss, and understand very basic things about the enzyme purification process. So we will going to have a detailed discussion on enzyme purification process, but in this first discussion, or you can say that in a first lecture, we are going to briefly understand what is enzyme purification, why it is important, and what are the basic steps. The different steps are there. What are basic steps it's involved in the enzyme purification process? Now, we first try to understand what is an enzyme. So we know about the central dogma. It says about DNA to DNA. This is the replication. It says about DNA to RNA, transcription. It says about RNA to protein. It is the translation. So while a gene it is being translated, it ultimately produces a protein. So what is a protein? It is basically the polymeric form of amino acids. Or in other language, you can understand that amino acids are the monomeric unit of proteins. Now, what is an enzyme? If this protein it has the catalytic ability, that means if it can perform, somehow it can perform any chemical reaction, then we call it as an enzyme that is related to different functions. So we know that we have thousands of metabolic reactions is ongoing in our body, in the you can say in the eukaryotic systems as well as in the prokaryotic system also. Right now, why the enzyme purification process is so important? By the term enzyme purification, we understand that we are going to have only one type of enzyme. That is only one type of enzyme it is going to be present. From the thousands of different enzymes of protein, we are going to isolate, we are going to purify the enzyme of our interest. Right? Now, why the enzyme purification process is so important? Because if you need to understand the function of an enzyme, we have to understand its structure. And if you want to if you're interested to understand or elucidate the structure of an enzyme, we need the enzyme in its pure form. So then only we can understand the structure of the enzyme, and once we have elucidated its structure, then we can easily understand the structure of the different structure and its function of the enzyme. So this is basically why the enzyme purification process is so important. So let's understand. Let's understand where exactly the enzymes is present. Suppose I'm taking the bacteria as an example. So the enzyme it's may present depending on its position. The enzymes it may be the intracellular enzymes or it may be the extracellular enzyme. The intracellular enzyme means those enzymes are present within the cell. It is not secreted; it is present within the cell. And extracellular enzymes are secreted outside to the cell environment. So if we culture a particular cell, it may be the plant cell, it may be the animal cell, it may be the bacterial cell. If it is the extracellular, then the enzyme it is going to be present in the growth medium, not within the cell. That's we need to understand it. Now, for the purification of an enzyme, there are several steps. Several chronological steps are there. But for the intracellular enzymes, first we need to break down. We need to break the cell. That is, we have to perform the cell lysis process. So thereafter, we have to perform other different steps involved in the enzyme purification process. So here we can understand, very critically you can understand that the purification process of those intracellular enzymes are little more complicated than those of the extracellular enzymes, because if you break down the cell within the crude extract, or we can say the cell homogenate or the cell lysate, we are going to have the different cell components along with our enzyme of interest. Now we let try to understand what are the basic steps, what are the different steps involved in the enzyme purification process. Now for the intracellular enzymes, definitely we have to perform the cell lysis process. We have to lyse the cell. We have to break the cell. So if you break the cell, then only the intracellular enzymes it is going to be present in the growth medium. So there are different techniques are there. It may be the physical methods, it may be the chemical methods, it may be the enzymatic methods. It may be we may use the high pressure, we may use the sound energy in order to lyse the cell. Now the choice of the cell lysis process, it depends on the type of the enzyme or type of cell we are dealing with. So depending on the cell and the type of enzymes, we prefer to use a specific type of cell lysis process. So do remember that this is for the intracellular enzymes. For the extracellular enzymes, we don't need any sort of cell lysis process, because the cells it is being secreted directly to the growth medium. After that, we perform the centrifugation process, mild centrifugation process. So in mild centrifugation process, we generally precipitate those cell organelles, cell membrane components or the cell wall components. It is being precipitated in the centrifugation process. We carried out a mild centrifugation process. So after that, we perform the salting out steps. So what basically salting out is that the enzyme of our interest, including the other protein and enzymes, it is going to be present in the soluble form. So in salting out process, basically what we do, we use a particular kind of salts which decreases the solubility of our enzyme of interest. So as the solubility decreases, so ultimately what will happen, our protein of interest is going to be precipitated. It is going to be precipitated. So we are going to isolate the precipitate where our enzyme of interest, including the other proteins and enzyme, it is going to be present. The salting out process, basically it decreases the solubility of our protein of interest. Right? So after that, we perform the fractionation process. So in fractionation process, we are going to have two different phases. First phase is the dialysis, and the second phase is the chromatography technique. So in dialysis, what we do, we use a semi-permeable membrane. We use semi-permeable membrane, cellulose-based semi-permeable membrane. It allows the diffusion of small molecules, not our protein of interest. That means our protein of interest, including including similar kind of protein and the larger protein as well as enzymes, it is going to be present within the dialysis bag, and that dialysis bag is made up of semi-permeable membrane. That membrane, it intact our protein of interest and other similar kinds of protein and a larger protein or enzymes. However, those small molecules, they diffuse to the semi-permeable membrane. So after the dialysis process, we perform the chromatography process. So in chromatography, we are going to have three different process. One is the size exclusion chromatography, second is the ion exchange chromatography, and the third is the affinity chromatography. So if you see the terminology, size exclusion chromatography, it relies on the size of the protein of our interest. In ion exchange chromatography, it relies on the charge that is present in our protein of interest. And lastly, affinity chromatography. It relies on the binding affinity, how strong the binding affinity between our protein of enzyme of interest and the ligand. It is embedded in the matrix, so it is relies on the binding affinity. So in enzyme purification process, basically, out of these three techniques, what are these techniques? Techniques means the size exclusion chromatography, ion exchange chromatography, as well as the affinity chromatography. Out of these three techniques, two techniques are used simultaneously. It may be size exclusion chromatography including the ion exchange chromatography. It may be ion exchange chromatography including the affinity chromatography. It may be the size exclusion chromatography including the affinity chromatography. Two of the three chromatography techniques it is used in the standard enzyme purification process. So up to this chromatography, when the chromatography process is over, we are going to have our pure enzyme of interest. It is in pure form, but that is present in a solution. That means water is present. So that means we, in order to do the packaging process, we have to remove the water. We have to remove the water, and the water is removed in the form of ice. So that this process, basically, very simply, if you understand, this process is known as the lyophilization process, or alternatively it is said as the freeze drying process. Water is removed in the form of ice. It is also called the sublimation process. Now, very critical understanding is that why lyophilization process it is used. The lyophilization process is used in order to maintain the activity of the enzymes. Suppose we have purified our enzyme, and this enzyme it loses its activity. But the critical understanding is that although we take care of all those preventive measures in order to maintain the enzyme activity, yet we used to purify the enzymes that is devoid of mostly it is devoid of its activity. So in this case, we use the different preventive measures so that we maintain the enzyme activity, and therefore in the lyophilization process, by using the lyophilization process, we tried our level best to maintain the enzyme activity. Done. So once the water is being removed, that means our enzyme of interest right now it is in powder form. So thereafter we go for the packaging process. So this is basically the steps, different steps it is involved in the enzyme purification process. So in my first lecture, or we can say the discussion, we try to understand why the enzyme purification process is important. I say that we need to understand the structure. So as we need to understand the structure is related to the function. So we need to have an enzyme in its purest form. Then we found that the enzymes are basically intracellular or it is extracellular. We understand the central dogma related to the expression of the enzymes. Then we understand the basic steps involved in the enzyme purification process: cell lysis, then it is the centrifugation, followed by the salting out, then fractionation. In fractionations, we understand that there are two different phases are there. One is the dialysis phase, second is the chromatography phase. In chromatography phase, we understand that out of the three different techniques, two techniques are simultaneously two techniques are used. It's maybe the size exclusion chromatography including the ion exchange chromatography. It's maybe the ion exchange chromatography including the affinity chromatography. It's maybe the size exclusion chromatography including the affinity chromatography. After that, the water is being removed to the process. It is a lyophilization process, and ultimately we are going to have the enzymes in its dried form, and those enzymes it is sent for the packaging purposes. In my second lecture, we will try to understand procedure or the steps that is involved in the different phases of this enzyme purification process. So today I'm going to conclude my lecture here. So you are encouraged to give your valuable suggestion as well as comments in my first lecture. Your suggestion as well as the comments are most valuable for me to improve my lecture series. What are the contents I should place here, and how should proceed my discussions, and what are the levels of discussions I should include here. So by saying this, I'd like to conclude my lecture here. See you again in my next part of our enzyme purification process. Till then, it's goodbye and take care, all of you. Thank you.